Hello and welcome to our seminar on surface plasma and resonance. I'm Philip Page, an application scientist at Reichert Analytical Instruments. Today I will cover the theory behind surface plasma and resonance, review some data examples, and look at the unique problem-solving aspects and features of Reichert's SPR instruments. First, a bit of our company's background. As you can see, we have been around for a while. Our heritage includes the design and manufacture of various optical instruments, spanning well over a century. Our entrance into SPR refractive index measuring instruments began in 1997 with a collaboration with Professor Carl Batt at Cornell University. We then introduced our first single-channel SPR instrument in 2001. Our first two-channel instrument came online in 2006, and last year we introduced our high-sensitivity SR7500 DC dual-channel instrument. Last year, we also became part of AMETEK's Ultra Precision Technologies division. Now I will proceed to talking about the principle of SPR. To induce the surface plasma and resonance phenomenon, a wedge of polarized light, spanning a range of incident angles, is passed through a high index of refraction prism under total internal reflection conditions. In this case, all the illumination light is totally reflected off the prism, and the reflected light is very intense across the entire reflected beam path. In SPR, a thin film of an electrically conducting metal is placed directly onto the prism in the form of a metal-coated glass chip. Gold is readily used because it is the most chemically inert metal that can give an SPR signal. The polarized light impinges on the gold chip at an interface between two different refractive indices, the gold chip itself and the medium directly above the gold surface, which is typically aqueous buffer. An electric field intensity or an evanescent wave is created as the light impinges on the chip. This wave penetrates into the gold surface and is absorbed by the free energy electron density clouds of the gold metal. This interaction generates oscillating electron density waves that are bound to the gold surface called plasmons. This resonance effect causes a reduction in the reflected light intensity with an energy minimum occurring at a specific resonance angle. This resonance angle is sensitive to changes in refractive index occurring above the gold chip surface. Surface plasma and resonance, or SPR. As was mentioned previously, the SPR phenomenon is brought about by shining a polarized wedge of light onto a metal surface that is at the interface between two widely different refractive indices. This incident light can interact with the electron constellations within the gold, causing a reduction in the reflected light intensity, seen as an energy minimum. Now, if we envision tethering a biological molecule, such as an antibody, to the gold surface, and then introducing its binding partner, the antigen will bind to the immobilized antibody and in turn cause a change in refractive index. This change in refractive index in turn causes the resonance angle minimum to shift to a different angle that is proportional to the mass of the bound material on the gold surface. In a SPR instrument, this angle change is converted to a response and recorded as a function of time, known as a sensorgram. SPR instruments measure very precise changes in refractive index, or in essence mass, at the surface of the sensor chip. The traditional units of measure is a micro-RIU, or 1 × 10⁻⁶ refractive index unit. 1 micro-RIU corresponds to a change in mass at the surface of 1 picogram per millimeter squared. Now, the RMS noise level of the Reichert SR7500 DC is 0.05 micro-RIU, or equivalent to a mass change of only 50 femtograms. This high sensitivity enables the monitoring of either low molecular weight molecules or very few molecules of higher molecular weights with great confidence. SPR can tell us many things about how two molecules bind or interact. Simple tests can quickly determine whether or not there is binding and rank the relative binding level of many compounds. More complex experiments are able to characterize rate constants, on and off rates, or kinetics. Data from experiments or responses reach equilibrium is used to calculate the equilibrium binding constant, or affinity. SPR can also be used to determine concentration, as well as thermodynamic constants, answering the question, why does the interaction happen in the first place? So we are able to fully detect, identify, and characterize based on the binding of one biomolecule or small molecule to another. This slide shows a typical SPR tensogram, a plot of instrument response versus time. The experiment starts with buffer flow over a biomolecule mobilized on the center chip surface. We then introduce or inject a binding partner or analyte in the flow chamber. As binding occurs, the mass at the surface increases. The instrument is monitoring change in refractive index or dielectric at the surface. The binding response normally follows an exponential growth curve. As more surface sites become occupied, the probability of future binding decreases, and the rate also decreases. The injection is then stopped, buffer is reintroduced into the flow chamber, and the dissociation or decay is allowed to proceed naturally. If the dissociation is complete in a reasonable time, usually minutes, a regeneration step is not necessary. However, if the rate of dissociation is fairly slow, regeneration is needed to completely dissociate the molecules, so the surface can be reused for multiple analyte injections. This regeneration step is accomplished by injecting a solution with either a high or low pH, high salt, or perhaps detergent. The trick here is breaking apart the interaction without denaturing the biomolecule mobilized on the surface. Once regeneration is achieved, baseline is reestablished and the surface can be reused for several successive analyte injections. Why are kinetics important? Mathematically, the equilibrium dissociation constant is defined as the off rate or the dissociation rate constant divided by the on rate or association rate constant. If we look at our plot of the on rate versus the off rate, we can see that interactions with vastly different on and off rates can have the same equilibrium dissociation constant or affinity. In this example, three completely different interactions all have a 10 nanomolar affinity. The lower right inset shows a slow on rate followed by a slow off rate, while the upper inset shows a fast on rate followed by a fast off rate. Rates are extremely important in determining how biological processes or pathways proceed. They are also a very important consideration in drug candidate selection. Here we see a conventional experimental approach. In this example, 7 concentrations of analyte are injected with a regeneration step in between. Usually the concentration series is rerun to ensure reproducibility. We typically try to bracket the expected equilibrium dissociation constant using 2 to 3 fold changes in concentration. For example, if the expected equilibrium dissociation constant is 10 nanomolar, the injections would be 2.5, 5, 10, 20, and 40 nanomolar. The associations and dissociations for each injection are then overlaid and normalized in preparation for curve fitting or data analysis. It is important that duplicate injections show the same response. Otherwise, it is likely the regeneration step is having a negative effect on the immobilized biomolecule. This is an example of what we call a kinetic titration. In this approach, increasing concentrations of the analyte are injected, followed by a dissociation phase in a single cycle. An advantage of this approach is no regeneration is required. This can greatly minimize assay development time by eliminating the need to find a suitable regeneration solution. This technique also makes an SPR assay possible when regeneration is denaturing the immobilized biomolecule. If the response data from each injection reaches equilibrium, the equilibrium dissociation constant can be calculated from a fit to a Langmuir binding isotherm, a plot of the equilibrium response versus concentration. In this case, we are using steady-state data as opposed to kinetic data to calculate the affinity constant. SPR can be used to measure the enthalpy and entropy of binding. This type of an experiment requires running the assay at 5 different temperatures. The data displayed here shows the binding of 4-carboxybenzene sulfonamide to the immobilized enzyme carbanic anhydrase 2 at 2 different temperatures, showing that the on and off rates are faster at high temperature. Enthalpy and entropy are related to the equilibrium dissociation constant through the Van 't Hoff relationship, as shown in the inset equation. Now, if we plot the natural log of the equilibrium dissociation constant versus reciprocal temperature, the slope is simply equal to negative enthalpy divided by our gas constant, while the intercept is simply equal to entropy divided by our gas constant. Entropy and enthalpy calculated from SPR data has been found to agree very well with data obtained from isothermal titration calorimetry experiments. Reichert SPR instruments are well suited to running experiments at different temperatures. Our instruments have an extremely wide temperature range, from 10 degrees Celsius to 70 degrees Celsius. This capability has enabled experiments at high temperatures using proteins from thermophilic organisms. SPR can also be used for concentration determination. This is quite easy if an antibody exists for the biomolecule that is being measured. The antibody is immobilized and a range of known standards are injected. The initial 10 to 20 second response is normally fairly linear. This produces a straight line when a response point at 20 seconds is plotted versus concentration. Points in the nonlinear parts of the response curves or dissociation can also be chosen and fit via nonlinear regression. Generally, a number of different time points are tried to determine the best fit. This next group of slides will provide an overview of Reichert's SPR instrumentation. Our SPR systems are analogous to component-based HPLC chromatography systems. They consist of a pump, an injector, a valve to direct flow over the two channels, and the SPR instrument or detector. The autosampler is capable of injecting from 2 384 position wall plates. The first component of the system, the syringe pump, is capable of a very wide flow rate range and accommodates syringes from various sizes, from 250 microliters to 5 milliliters. The pump features very accurate pumping down to 100 nanoliters per minute. The wetted materials of the pump are glass, Teflon, and ceramic. The pump is controlled via our system software. Refills and flow changes can all be incorporated into easily set up sample run tables. The next part of the system is the injector. This can be an autosampler, semi-automatic injector, or both. The autosampler can inject from 296 or 384 ball plates, or 248 or 12 vial trays. It has sample cooling down to 4 degrees Celsius to preserve thermally sensitive compounds. It allows for the automatic execution of experimental protocol for 24 hours of unattended use. The semi-automatic injector has a much smaller footprint as compared to the autosampler. It is also computer-controlled for injection times. It also provides injection flexibility. It can be used to immobilize a ligand which may coat the fluidics, or it can be used in series with the autosampler to inject an inhibitor, followed by the analyte under study. Performance of our feature detector, the SR7500 DC, is due to a homogenized feedback-controlled LED light source, an extremely sensitive linear photodiode array detector. This detector has large pixels to enhance light gathering capability. It has extremely low dark current and a 100,000 to 1 dynamic range. The root mean square or RMS noise of this detector has actually been measured at 0.05 micro refractive index units at 25 microliter per minute flow rates. The refractive index range of this detector is from 1.32 to 1.42. It can be extended to 1.52 using a high index glass slide. The instrument can detect 100 Dalton analytes. It incorporates a sapphire prism which is virtually indestructible, and a temperature control from 10 degrees Celsius all the way up to 70 degrees Celsius. The specifications provide an analyte concentration range of 10 to the minus 4 to 10 to the minus 12 molar. This slide shows a 5-minute reference extracted baseline segment. The flow rate was 25 microliters per minute and the temperature was 25 degrees Celsius. The RMS noise of the segment is calculated at 0.05 micro RIU.